Now, happily, many of you already know our speaker. Dr. Mandal is an associate professor at Roseman's College of Pharmacy. In 2019, he was awarded a Fulbright specialist appointment by the U.S. Department of State's Bureau of Education and Cultural Affairs and World Learning. His expertise is teaching and research in immunology. He received a PhD in science from the Jadavpur University in India and did postdoctoral research at the University of Chicago and the University of Michigan. He was also involved in biodefense research at Virginia Tech. Dr. Mandal has given invited talks in the US and abroad and chaired scientific sessions. He has mentored many students, fellows, and faculty, and is professionally associated with numerous pharmacy, education, research, and accrediting organizations. Dr. Mandal is also a member of the American Association of Immunologists and the American Association for the Advancement of Science. And now it's our privilege to hear from Dr. Mandal.
Thank you, Brenda. I really appreciate, and I appreciate some of you joining us this evening. Little update on my status, meaning I received a full professorship appointment promotion as of beginning of July 1st, but nonetheless, I'm the same person who taught you immunology, and I am very much invested in COVID-19 infection, vaccination, and I helped along with you for COVID-19 immunization through Roseman University's immunization clinics. So today we are going to talk about COVID-19 vaccines and variants and what are in store for us looking ahead. To begin the seminar, I will first briefly introduce last year's seminar, which was given to you. Some of you may have been present on this platform last year, August 13 in 2020, where we talked about an emerging virus infection, coronavirus infection, SARS-CoV-2, various molecular aspects of this infection, pathophysiology, host immunity to the infection. And on that platform, meaning last year, we ended up with talking about vaccine development approaches. That's highlighted in yellow on this slide. At that time, vaccine development were in full swing, but there were no available vaccines against COVID-19. Fortunately, moving forward this year, beginning of this year, we have several vaccine candidates available and received emergency approval or authorization from FDA. And very recently, Pfizer vaccine got full regular authorization from FDA. Taking all this information together, we'll talk about COVID-19 vaccines and vaccination efforts, vaccine safety, and definitely we'll mention about the COVID-19 variants that are causing concerns, particularly Delta variants. What are the vaccine-induced immune protection against this variant? Recently discussed vaccine booster doses and what we have learned collectively over the year, last year and through 2021, and how close are we going back to normal, or what will be the new normal for us? I always begin with a little bit of philosophical approach in my talk. This is a quote from Albert Camus on the description of the plague. What is notable here in the last line, which states, there have been as many plagues as wars in history, yet always plagues and wars take people equally by surprise. Currently, we have a plague on hand, and in a century, this is the first pandemic we encountered in our lifetime. And this time, it is coronavirus infection. And this is the beginning. We have seen this slide before, if you have attended my last talk last year. But summary is that if recent US intelligence reports into the origins of coronavirus pandemic remains inconclusive. A team of US scientists and researchers went into China. They gathered all the information, talked to people, and this report came out in August 2021. And the origin of coronavirus pandemic remains inconclusive. But most likely a bat somewhere in China or Southeast Asia got the virus into animals, and that animal-specific coronavirus infection gotten into human being in wet market of Wuhan in China. So coronavirus infection triggers a respiratory disease. This is a member of Coronaviridae family. From this family of viruses, we had MERS, we have SARS-CoV. They infected us in 2012 and previously, but current infection is known as coronavirus infectious disease 2019, or in short, COVID-19. It's a single-stranded RNA virus. One of the notable features of the structural aspect of this virus is spike protein. This spikes out of this viral structure give this virus known as coronavirus. And this is a family of virus that has infected us before, and current coronavirus infection also carries these spike proteins. What this spike protein does, it binds to the H2 receptor in our body, especially in the lung nasal epithelial cells, and H2 overexpression in our nasal and nasopharyngeal passages may be correlated with enhanced transmissivity and viral reservoir in us, meaning in infected individual. This slide shows the SARS-CoV-2 entry and its life cycle. It begins with viral particle binding to the H2 receptors. Then there will be RNA replication, transcription, translation, and assembling of the new viral particle. Ultimately, a variant, new viral, mature variant, or new viral particle will be released. And when that infected individual sneezes, coughs, another individual who may be hitherto uninfected, if they gets exposed to this released viral particle, then they get infection. That's how this SARS-CoV-2 infects a particular individual, and the infection is transmitted into another uninfected individual. Down below, it shows that there are many structural aspects of this coronavirus, particularly spike proteins and its domains, such as receptor binding domain and NTD domain, that are very essential into the binding of SARS-CoV-2 into H2 receptor structures. And if we come up with an intervention antibody specific for this spike protein that could block this viral binding, then we could prevent the infection. And that is exactly achieved through vaccination or immunization. On the right-hand side, lower right-hand side of this panel shows that following that thought process, many monoclonal antibody drugs have been developed. Those monoclonal antibodies originally isolated from an infected individual who generated spike protein specific antibody, and those antibody clones now are generating life-saving antibody drugs that are used to treat an infected individual. Little bit of COVID-19 infectivity and mortality. We know that COVID-19 jumped species, and that has increased its vulnerability to infection and disease development. There are many variants, such as alpha, gamma, or very recent Delta variant, all known to increase infectivity. Particularly, Delta variant is implicated in increasing the infectivity by 60%. It triggers mortality rate very high, about 1%, whereas seasonal flu mortality rate is about 0.1%. So coronavirus infection comes with a 10 times higher mortality rate compared to influenza infection. R naught value, meaning one infected individual, how many others can get infected from one infected individual, meaning the spread of infection, rate of spread of infection is defined as the R naught value. That ranges between 2 and 2.5, which is very high. Probability of fatal disease is highest in elderly people and deadlier thus in older people. Asymptomatic infection prevalence is about 45% of the individuals infected with these viruses, this particular various variants and the original strains, whereas asymptomatic patients can transmit 75% level up to 14 days after getting infection. But since they are asymptomatic, they would not even know that they are transmitting the virus and they are infected. All these informations taken together tells us one very simple information, that if someone wears a mask, even when they are fully vaccinated, that cuts down the transmissibility and infectivity in them and into other individuals. Clinical features of COVID-19, it impacts older males about 50-year or higher age group, but with Delta variant, that age group is now lower. The prime age group for Delta variant infection mortality is about 40 to 50. Sure, that's something different compared to the original virus strain. Comorbidities for infection, obesity, high blood pressure, cholesterol, diabetes, kidney and heart disease. Most clinical manifestations are fatigue, muscle body aches, fever, chill, cough, loss of tastes and smell, and sometimes shortness of breath, nausea, vomiting, and diarrhea. Taken together, this virus infection may trigger severe cases, and one may develop acute respiratory distress syndrome or ARDS. Immunologically, it triggers exhaustion of the white blood cells, leukocytosis and lymphopenia, and triggers inflammatory biomarkers at a high level, such as ALT, LDH, and D-dimer, including reactive protein and ferritin, which are significantly higher in severe cases compared to the more moderate cases of the infection. Also, in severe infection, various inflammatory cytokines such as interleukin-1, IL-6, TNF-alpha, and IL-10 significantly increased in severe cases and markedly higher in moderate cases. So with that clinical information and background, let's take a look at current US COVID-19 cases, deaths, and vaccination. This data is absolutely updated as of this morning's new data. As of now, there are about 43 million confirmed cases in US, and unfortunately, over 680,000 deaths in US. 7-day average of new cases is 122,000, which is a good news because it's down about 17% compared to last week. But the bad news is 7-day average new deaths are spiking up 8%, which is now over 2,000 individuals. 55% of US adults and who are eligible to get vaccinated are fully vaccinated, and global totals of infection is 232 million cases with about 5 million deaths. And this data on the right-hand, these graphs, right-hand side panel shows all this information. And if you follow the green line, that's the US data. As of right now, it spiked up a little bit and fortunately going down. This information shows the SARS-CoV-2 infection and illness timeline. Without going into the detail, it shows that median incubation period of this virus infection is about 5 days, whereas peak transmissibility is half a day before one shows symptoms of infection. Once again, this specific information demonstrates the importance of wearing a face mask, because if one wears a face mask, they would not be able to transmit the viral particles, even when they do not know that they already got the virus infection in them. Median duration of the fever is about 4 days, and peak nasopharyngeal viral load that can be detected with RT-PCR or rapid antigen testing between 3 to 5 days after infection. All these numbers are different in Delta variant, and I will talk about these numbers when I go to the Delta variant specific information. And right-hand side diagram shows the phases of COVID-19: viral phase, then cytokine storm, then becomes a coagulatory phase if it happens, and it ends with late hyperinflammatory phase. There are many molecules that have been associated with the infectivity, transmission of the viral infection, particularly jewel crown of coronavirus infection is S protein. And this is the molecular structure of S protein. And there are various domains, and without going into the details of these domains, it's sufficient to mention that these are not only the targets for vaccine development approaches, but these are also targets for antiviral drug development. approaches. And on the top upper panel, it shows that out of the original strains of coronavirus infection, we had earlier, these are the beta coronavirus family of viruses. We had MERS, we had SARS, then the original COVID-19, and now we have Delta, Gamma, and Alpha variant. Particularly this Delta variant is prevalent worldwide, found in 135 countries of the world. And in US, this is the variant of concern. So what are the mutations and variants? Errors in viral replication or duplication leads to mutation, and viruses with these mutations are called variants. There are many mutations observed from the very beginning of this SARS-CoV-2 or COVID-19 infection, and these are the notable mutations. Particularly E484K is found in Delta variant, and many other mutations led to Alpha, which originated in UK, Beta in South Africa, Gamma in Brazil, and current Delta variant was originally developed in India. All these variants are called variants of concern. Why? Because these variants increase transmissibility, triggering higher viral load, shorter incubation period, higher degree of pathophysiology, pathogenesis, and disease severity. Both Alpha and Delta variants showed increased infectivity and disease severity and hospitalization compared to the original strain. And mutation in spike protein somewhat compromised the vaccine effectiveness and vaccine-induced immune protection. Very recent, as of last month, there was a model utilized to forecast recent proportions of circulating variants in US. What it shows that nationally, over 98% new infections currently caused by Delta variant, which originally developed in India about end of last year. According to WHO, the Delta variant has spread over to 135 countries all over the world. Why we talk about these details of this variant and their prevalence? Because this type of modeling help us estimate the current prevalence of the variants based on the genomic surveillance, so that not only we can monitor the emergence of the variants, its infectivity, but if we have those data early on, then we can put the effective protective measures such as various increase in vaccination, treatment facilities, various oxygenation measures, and etc., ICU preparedness to mitigate the emergency situation. Attributes of these variants of concern, VOC, essentially they increase transmissibility, increase disease severity, increase viral load, and comes with a shorter incubation period. Taken all these informations together, realistically what it does, it decreased neutralization abilities by the antibodies that one may have generated due to previous infection or due to COVID-19 vaccination. Thus, reduced vaccine-induced protections are observed due to these variants of concern now circulating all over the country. Particularly for Delta variants, some notable information came up very recently. This Delta variant was generated due to a mutation into the furin cleavage site of the spike protein that increased the efficiency and quantity of the virus that enters an infected cell. One particular study estimated that viral loads are up to 1,000-fold higher compared to the Alpha or Beta variants. Similar viral loads were found in vaccinated or in unvaccinated individuals if these individuals get infection with Delta variant infection. And for Delta variant infection, incubation time is shorter, about 4 days, shortened by 2 days, where original infection has an incubation period about 6 days. Risk of hospitalization doubles almost with the Delta variant versus the Alpha variant. And those in hospital were likely to suffer from more severe symptoms if that individual is infected with a Delta variant. So taken together, this virus is Delta variant is more deadly compared to the original strain, because not only it triggers 1,000 times more viral load, it also somewhat compromises the vaccine-induced immune protection. But if the individuals are vaccinated, most likely they do not need to go to a hospital, meaning they would not suffer severe infection, hospitalization, or death. Before I go into the vaccines and vaccine-triggered immune protection, let's review a little bit about immune responses that are generated due to COVID-19 infection naturally in us. When we get infection, be it a virus infection or bacterial infection, we trigger two different types of immune responses. One is innate immunity, that's generated via monocyte, macrophage, neutrophils, and natural killer cells. That comes up very early on in the course of viral infection or any infections. Subsequent to that, adaptive immunity kicks in. That takes about 5 to 7 days. And this adaptive immunity particularly triggers B-cell activation-induced antibody responses and T-cell activation, such as CD4 and CD8 T-cell activation. Taken together, through innate and adaptive immunity, viral infection is usually controlled. What remains after virus infection, long-lived memory B cells and T cells that provide protection against future infections. COVID-19 vaccines, luckily, have been shown that they generate good antibody responses and to some degree T-cell responses, along with immunological memory towards vaccine antigen. We will talk about this data as we move forward with COVID-19 vaccines and variants. So early on, last year, when we learned about this new infection developing all over the world, scientists and clinicians went into work. What they did is a remarkable feat of scientific and clinical achievement. Usually, a vaccine, particularly a new vaccine, takes about 10 or 15 years to come to the market. Right from the conception of these vaccine development approaches, one needs to do preclinical analysis in the lab, then phase 1, phase 2, and phase 3 clinical trial, then regulatory review, vaccine approval, and manufacturing starts. All these things takes about 10 years at least. And even 10 or 15 years is not a guarantee of bringing a new vaccine to combat a disease, particularly infectious disease. But this time we got lucky. Scientists and clinicians brought COVID-19 vaccine at a record-breaking speed within an year or little over an year, I would say. What are these vaccine and vaccine development platforms? These were inactivated, weakened virus vaccines, or viral vector containing replicating or non-replicating COVID-19 RNA sequences and DNA sequences. And there is also a protein-based vaccines and virus-like particulate matter or antigen vaccines. And this is a little old data. Old means last year's data. At that point, number of vaccines in the development, viral vector vaccines and protein-based vaccines were leading the chart. And down below, it shows that upon immunization with COVID-19 vaccines, they trigger essentially good antibody responses through B-cell activation, and to some degree T-cell activation also occurs. And these are the leading COVID-19 vaccine formulations currently we have in the market. mRNA-based vaccines, such as Moderna and Pfizer-BioNTech. These two companies came up with a spike protein-specific mRNA formulation in 4 different kinds of lipid encapsulation. That was pretty successful effort. Whereas adenovirus vector-mediated vaccine development occurred with AstraZeneca and Johnson & Johnson, Gamaleya, and CanSino Biologics. So these mRNA and viral vector vaccines are currently the leading vaccine candidates all over the world. But DNA vaccines being developed by IonoBio very recently, means in a next few months, we hope that this vaccine will be first introduced into the Indian market and worldwide. Whereas protein-based vaccines developed by Novavax, a US company that also uses traditional protein-based vaccine approach targeting S proteins that will be also released worldwide, perhaps beginning somewhere else outside in US. And another type of vaccines currently used, particularly in India and some few other countries, is killed virus vaccine. Covaxin, a Indian company developed, such as Bharat Biotech, that's the Indian company, brought in the Covaxin in India. And in China, Sinopharm, that's also utilized this approach, killed virus vaccine approach. But no matter what is the vaccine formulation, overall, all these vaccines are very safe, efficacious, and protective against SARS-CoV-2 COVID-19 infection. And this slide shows the data of this vaccine efficacy. Without going into the details of these numbers, reading out, we can see that overall, Pfizer, Moderna vaccines are about 95% protective against infection, whereas Johnson & Johnson one-shot vaccine slightly lower in protection, 66% to 85% protective against severe infection. AstraZeneca vaccine that utilizes adenoviral vector platform, it's about 70% efficacious, but this vaccine is not utilized in US. Whereas many other vaccines such as Sputnik V, which is a Russian vaccine, Bharat Biotech vaccine, Indian vaccine, Sinopharm, Novavax, all are in the range of 80 through 90% efficacy. Therefore, take-home message is that all the COVID-19 vaccines available all over the world are highly efficacious, highly protective against symptomatic COVID-19 infection. This slide shows what's really inside an Moderna, inside an mRNA vaccine that's been developed by Moderna and Pfizer BioNTech. The intuitiveness and the ingenuity, I should say, of these two mRNA vaccine is that when one particular scientist in UK modified the uridine nucleotide into pseudo-uridine, that led to the stability of these mRNA sequences, spike protein specific mRNA sequences within a lipid encapsulation. Meaning mRNA sequences are notoriously unstable molecule. But the stabilization occurred when the uridine was modified into pseudo-uridine. And that's the ingenuity of developing these lipid-mediated mRNA vaccines developed by Moderna and Pfizer. This slide also shows the efficacy of various vaccine formulations such as Pfizer, Moderna, J&J, AstraZeneca, and Novavax. Without, again, going into the detail, the highlighted area such as this, overall, all these vaccines are about 94, 95% efficacy against protecting against severe infection. AstraZeneca vaccines varies, efficaciousness varies slightly compared to Moderna and Pfizer vaccine. It's about 70%, so is Johnson & Johnson vaccine, but nonetheless, they are highly effective against severe infection and prevention of hospitalization and death. And that information is shown below. COVID-19 death due to UK variant, South African variant, Brazilian variant are highly protected with immunization with Pfizer BioNTech at 100%, Moderna, J&J, AstraZeneca, and Novavax vaccine, meaning these vaccines are not only protective against the original strain, they are highly protective against Alpha, Delta, and Brazilian P1 variant. This is a very new data that shows total number of vaccines administered against COVID-19 and vaccine doses in US as per this morning's CDC report. Vaccination in US really took off since mid-December last year. And around March through May, it was peaked. Then unfortunately, vaccination rate slid down and went to really very low around June, July. And it picked up once again through massive news informations and about the vaccine safety efficacy from various sources. But still, currently, the vaccination or immunization rate is not up to the standard or satisfactory level. It's slightly going down. And which vaccine has the greatest global reach? Oxford's AstraZeneca vaccine, Pfizer, BioNTech, and Moderna leads the pack. These three vaccine formulations are utilized many countries all over the world. These are the number of the countries, like 110 for AstraZeneca vaccine. Pfizer BioNTech vaccine is utilized in 78 different countries, and Moderna vaccine is 36 different countries. Taken together, these are the leading three vaccine formulations all over the world. Little bit of vaccine efficacy, effectiveness, protection, and breakthrough infection. We hear all these terminologies all the time through TV information and information bombardment, but what really these terminologies mean? Vaccine efficacy refers to how the vaccines will perform in ideal condition, meaning in a clinical trial. Effectiveness refers to how the vaccines will protect vaccinated individuals at large worldwide. And when we say that one vaccine is 80% effective or efficacy with 80%, that means 80% reduction in infection. Top panel shows that this is the placebo group, meaning they did not receive the vaccination. Down below, this is the vaccinated group. After a certain time point, clinicians will track these individuals in the vaccinated versus placebo group. And here we see these two red individuals got infection, but many other individuals in placebo group, unvaccinated group got infection. The difference is about 80% between vaccinated versus unvaccinated group, and that refers to the vaccine efficacy. As I mentioned before, Pfizer, Moderna, AstraZeneca, and J&J vaccines all stimulated strong antibody responses and T-cell activation and prevented COVID-19 infections. Pfizer and Moderna vaccines work in real-world conditions as per CDC, over 90% level, effective at full vaccination, preventing infections, including asymptomatic infection. Both these Pfizer and Moderna vaccines are effective in pregnant and lactating women that triggered IgG, IgN, IgM class of antibody responses. And it has been observed that vaccine-induced antibody found to be greater in the natural infection-induced antibody responses, meaning a vaccinated individual triggering higher level of antibody responses compared to a naturally infected individuals. Both Pfizer and Moderna are currently working on developing multivalent vaccine formulation, adding the variant-specific sequences. Immune correlates of protection is very difficult for any vaccine formulations, meaning what really protects an individual who has been immunized against an infection or who has suffered a natural infection and survived. We know that B cells, T cells, all are active towards neutralizing the infection and mitigating the infection, bringing normal health in the infected individuals. But what exactly works, that oftentimes is very difficult to determine, that's known as immunological correlates of protection. We still do not know, after 40 years of HIV/AIDS research, what is the immunological correlates of protection against AIDS. Similar fact is true for COVID-19 infection. Perhaps it's a little early, but taken together, B cell and T cell responses and cytokine release and killing of the infected cells, particularly by cytotoxic T lymphocytes and triggering of memory B cells and T cells, that provides long-term immune protection and refers to the longevity of the immune protections. All these are important aspects of COVID-19 vaccines. Before we go into the variants and variant-specific immune protection due to vaccination, let's look at the real-world vaccine protection data. There are many data available currently, such as Israel data, which is the strongest data worldwide that shows that upon receiving 2 doses of Pfizer vaccine, individuals are 95% protected against infection and 96% protective against hospitalization. critical illness or death. Delta variant as the dominant variant strain, and 61% of the individuals in that country was vaccinated. So vaccine effectiveness was over 90% in documented infection and 84% against symptomatic infection in transplant and immunodeficient patient population, which is a strong testimonial of the vaccine protection against Delta variant. Kentucky data, Mayo Clinic data in US also refers to the similar level of numbers, around 90% of protective efficacy with these vaccines. Particularly Mayo Clinic data shows that pre-surgical screening of 9,000 patients who received full vaccination, which was a requirement for the surgery, that was associated with 80% lower risk of testing positive for COVID-19 and 97% effectiveness for preventing symptomatic disease, severe disease, and death, and 94% efficacy against asymptomatic infection. Similar data observed in India, Christian Medical College, CMC Vellore, which is an over 100-year-old hospital, very reputed hospital in India, southern part of India, that tracked its healthcare workers who received 2 doses of Indian vaccine. They were protected as 65% against infection and 77% protection against hospitalization, whereas who did not receive vaccination, they were infected at a much higher rate. Little bit of adverse reactions. Yes, there were adverse reactions, but those were very, very minor. About 1 in 10 individuals are known to suffer mild side effects such as tenderness, pain, warmth, itchiness, bruising at the injection site. Also, many patients suffer from chills, joint pain, headache, and nausea. Serious side effects was observed in some of the individuals with AstraZeneca vaccination protocol that included blood clotting, which is not a good news, but the frequency is about 1 in 100,000 individuals. 90 patients out of 34 million vaccinated with AstraZeneca vaccine formulations experienced blood clots in brain. Unfortunately, 18 died. But correlation of this 18 out of 34 million death is a correlation, but it's not causation. There is a difference between causation and correlation in clinical terminology. The mRNA vaccines are associated at a rate of 3 to 5 cases of severe allergic diseases per 1 million doses. And majority of these individuals who showed severe allergic diseases, they have a history of allergy. So although there are unfortunate deaths in vaccinated individuals, particularly with AstraZeneca and some J&J vaccines, because these 2 vaccines employ or come up with the same adenoviral vector platforms. The suspicion went into the platforms rather than vaccine antigens, but there are some very few unfortunate deaths. But the question is that, wouldn't there be 18 deaths even in 34 million unvaccinated individuals anytime if we take the history? Perhaps answer is yes. Therefore, correlation is not causation. And rightfully, COVID-19 vaccine complications are dwarfed by the risks of virus infection. A very recent British Medical Journal report analyzed over 29 million vaccine data 28 days after the first dose of mostly over 40-year-old compared to 1.8 million virus infection in many other individuals. It found that every 10 million people vaccinated with the AstraZeneca vaccine, an extra 107 would be hospitalized or die from thrombocytopenia, which is nearly 9 times lower than the risk of actual infection. About extra 66 individuals would be hospitalized or die from blood clots in veins, but that was nearly 200 times lower than the risk following a natural infection. For every 10 million vaccinated with Pfizer vaccine, it found 143 extra strokes, but that was nearly 12 times lower compared to the risk following a natural infection. And this slide shows some really very interesting data. risk of adverse side effects in AstraZeneca vaccine compared with other risks. Being hit by lightning, 1 in million. 11 in million will suffer serious harm due to AstraZeneca vaccination. Dying with coronavirus infection, 23 in 25-year-old category or 800 in 55-year-old. Due to accident, that number will be 110 and 180. Dying due to road accident, 38 and 23. But serious harm due to vaccination is only 11 or 4 per million. And that's the efficacy data, safety data, due to this coronavirus vaccination. Let's take a look at the SARS-CoV-2 variants and vaccination. All these variants, alpha, beta, gamma, and particularly current Delta variant, poses a risk of enhanced infection, transmissibility, and may compromise vaccine-induced protection to a certain extent. Because Delta variant has a shorter incubation period, comes up with higher viral loads compared to the original strain. A recent study shows that Delta variant makes up over 80%. The number is actually a little higher. Now it's over 90% of the new infections in US. But still, vaccination protects us at this level. Percent efficacy against variants with symptomatic disease. If someone got Pfizer, AstraZeneca, or Moderna and J&J vaccine against Delta variant, these are the protection numbers against symptomatic disease, meaning 88, 76, 72, or 67% protectiveness if someone received any of these vaccination. And percent efficacy against variants with hospitalization, they're highly effective, over 90%, 96% with Pfizer vaccine or 96% with Moderna vaccine or J&J vaccine. Folks are protected. If they received the vaccination, they would not go to the hospitalization, or certainly they would not die. At the max, they will have some mild infection, and with homestay, they can recover very well. And these are the data, again, points out to the fact that all these vaccines are highly effective and protective against severe infections and death. This is a little older data during April 2021, CDC data. At that point, it tracked about 87 million fully vaccinated individuals in US, and out of 87 million, there were only 88 deaths. Asymptomatic infection was in 2,000 individuals, and about 500 individuals out of 87 million required hospitalization. And that's the strength of this immunization and vaccines. And that brings to the variants and vaccine efficacy. Taken together, mRNA vaccines by Pfizer and Moderna, AstraZeneca vaccines are all highly protective against Delta and Gamma variants. Both Delta and Gamma variants required between 3.5 and 10 times much higher level of antibody responses that would neutralize regardless of the antibody origin, maybe due to vaccination, infection, or antibody drug treatment. Meaning, if someone is infected with this variant, and if those individuals are vaccinated, they are still highly protected, although higher level of antibody responses are required to clear up both Delta, both Gamma and Beta variant, which is also true for Delta variants. Because Delta variant reduces vaccine-induced immune protection to significant level compared to the original strain, although, and yet, if one is vaccinated, they are highly protected against hospitalization and death. Is there a booster dose required? Yes. All vaccines, almost all, I should say, require booster doses when immune responses goes down. And it has been observed that Pfizer vaccine's efficacy dropped to 84% from 95 or 96% after 6 months, and decline was calculated about 6% every 2 months. Therefore, it has been approved by FDA very recently in the last few days that booster doses will be required for individuals over 65 years of age and older, and particularly 18 through 64 years of age who are at high risk of contracting severe COVID-19 infection, These individuals may be healthcare workers, physicians, pharmacists, nurses, and emergency workers, or elderly care facility workers. So all are required to get a vaccines after 6 months of original vaccination. Booster doses for Moderna and J&J vaccination, also this approval process in the works, and very shortly we might hear from FDA that both Moderna and J&J vaccine recipients will also be helped with 1 booster doses. Before I go into the very last few slides of my presentation, let's talk about a little bit of vaccine hesitancy and what full approval of COVID-19 vaccines and booster dose means. There are vaccine hesitancy observed all over the world, including US. A Kaiser Family Foundation survey shows that US adults, about 1/3 of them surveyed, they're not yet ready to receive vaccination. 16% said that vaccine was too new or unknown, not tested enough, while 50% of the unvaccinated are on wait and watch list, meaning let's see what happens on that mode. Although FDA announced both Pfizer and Moderna booster doses for solid organ transplant on August 18, and FDA also authorized full approval of Pfizer vaccine on August 23, still the vaccination did not pick up significantly. Nearly 26% of COVID-19 patients recently reported nationwide in children. So eagerly, we are waiting from FDA, Moderna and Pfizer vaccine approval for 5 to 11-year-old, perhaps coming up in next few weeks. To understand the vaccine hesitancy and etc. in young individuals, I, along with few high school students at Green Valley High, recently did a research study learning vaccine hesitancy and their thought process in young adults in the high school students. And this was presented at a national meeting, AACP national meeting, and also at Roseman Research Symposium there. Children, particularly under 12, are particularly vulnerable due to COVID-19 infection. This is the new, really disturbing trend nationwide. There are many reasons for that. So again, we are eagerly awaiting Pfizer and Moderna vaccine approval for 5 to 11-year-olds to mitigate that. Finally, I will end this presentation where we are heading. The take-home message is that no one is safe until everyone is vaccinated. And this data shows essentially many of these influential and affluent Western countries, such as US, UK, Canada, Australia, European countries, they hold more vaccines than their entire populations, such as Canada holds 5 times more. US holds twice as many vaccines that can immunize the entire population. UK holds over 3.6 times more, etc. And there is a tale of vaccine inequality all over the world. High income countries holds the major vaccine supply chain all over the world, whereas upper and lower income countries are less fortunate to have available vaccine doses. And the data at the below shows the vaccine doses per 100 people in countries with highest total of vaccination, leading by UK, followed by US, Turkey, France, Germany, leads the chart. Looking ahead, what might happen? Coronavirus would struggle to max out both transmission and immune evasion at one go, meaning there is very early indication that even the Delta variant might lose its bite, meaning they are not so much successful in triggering infection, because people infected with the Delta variant are living in these individuals who survived with high antibody titers. Also, these vaccines are highly effective against these severe diseases and deaths. There is probably a limit to just how bad SARS-CoV-2 can get, because vaccine recipes can be tweaked to accommodate the new variants, and that can control the unabated transmission. But to achieve that, we need to increase the vaccination rate. And finally, viruses are infinitely cannot go through the mutation, and sometimes to keep themselves in contention, meaning in the business of infection, they must sacrifice, meaning their infectivity goes down. Perhaps current surge is likely to die down by Thanksgiving this year. This is a very, very early data that came out in last few days, but these are the predictions. Thus, COVID-19 will perhaps remain as endemic diseases like seasonal flu, and promising antiviral drugs, vaccines in clinical trials, and particularly antiviral drugs in clinical trials, results are awaited fall through winter, particularly various drug manufacturers such as Merck and Company coming up with new antiviral that might mitigate the new infections in individuals. And with vaccinations, nearly 100,000 more COVID deaths is projected, not with vaccination, but without vaccinations, 100,000 more COVID deaths in US projected between August and end of this year. The death toll could be cut in half if nearly everyone wear a mask and everyone takes a vaccine. And it's projected that vaccines so far prevented over 3 million new infections, 0.9 million hospitalization, and 128,000 deaths. Ultimately, our behavior will determine if, when, and how the current Delta wave will subside and pandemic will be controlled. So the transition to pandemic to endemic COVID-19, perhaps in the way. Realistically, the risk will be much smaller than it is right now with Delta wave, but it will never be gone, meaning COVID-19 infection, Delta variants will perhaps be contained in specific areas in the country. Therefore, from pandemic, it will be endemic infection in certain geographical area, and it will be a manageable infection. With a herd immunity, higher vaccination, better treatments, we might reduce the risk of COVID-19 further. The experience may also prompt us to take all respiratory viruses more seriously, leading to lasting changes with particular protective measures such as mask wearing, ventilation of the closed spaces such as office spaces and schools and etc. So we have learned quite a bit, but still effective early testing, quick results, tracing, quarantining, that would break the chain of virus transmission via physical distancing, mask wearing, and facility closure if necessary. But we need a better and comprehensive test, treatment, and vaccination guideline. Need to vaccinate enough people to trigger community or herd immunity. There is a need for central and uniform public health policies, which is seriously lacking right now in US, particularly in certain southern states where lower vaccination is triggering higher infectivity and deaths compared to the other areas of the country where vaccination rates are higher. There are public health measures are more effective, such as distancing, mask wearing, and etc. We need to know the longevity of vaccine effectiveness, and we have to encounter and overcome the vaccine hesitancy and vaccine inequality in certain populations in the country. Therefore, public awareness, education in infection prevention and vaccination, increase in vaccination will lead to mitigate COVID-19 infection. Mental health is an issue due to pandemic lockdown and isolation. We need to understand and do things about it. We need to learn long-term effects of COVID-19. That's a dark horse in whole saga of COVID-19 pandemic. Will COVID-19 remain as a community-based endemic infection? Perhaps the answer is yes, but we need to take COVID-19 vaccine every year like seasonal flu. And will there be universal vaccines for coronavirus infection and maybe seasonal flu, which is a coronavirus family of viruses, combined with COVID-19? Things are in the works, particularly along this line, combined seasonal flu and COVID-19 vaccines. Finally, $1 million question, will vaccination give our old life back? perhaps not to the full extent right now with the current vaccination rate, which is stands still around a little over 50% nationwide. Therefore, we still need to continue surveillance testing, genomic sequencing, isolating, tracing, quarantining efforts, and effective caring for the infected individuals. We need to follow the public health guidelines and measures universally all over the country to mitigate the uptick in the Delta variant infections. Human behavior more than the vaccines will determine how soon we can go back to normalcy. Unless a majority, which could be about 70% in a country and worldwide, are vaccinated, that would lead to develop a herd immunity. We really can't declare a victory. Therefore, access to tracing, genomic sequencing, and treatment will lead us or help us back to normalcy, but will greatly depend where one lives. Particularly, human behavior will be the pivotal point in determining the course of pandemic through endemic stage of the infection. Thank you for your attention. And at this point, I will take your questions.
Thank you, Dr. Mandal. I have one question. I know you recently traveled and you were wearing a K95 mask.
Yes.
Given the 1,000-fold viral load from the Delta variant, do you recommend anyone who's traveling on a large public vehicle wear a K or N95 mask?
Yes, so that's a good question. So recently I traveled to India to visit my family back home. It was a over 12,000 mile flight across the globe. Change of 3 airplanes. Longest flight was over 14 hours from San Francisco to India, Delhi in India. Anyways, so all this time, over 30 hours for the duration of my travel, I wore a KN95 mask. And at one point, I had regular surgical mask underneath the KN95. What it does, scientific research has shown that a mask such as KN95 mask is the best in protecting against transmission of the viral particle from one infected individual to another individual. That has been scientifically documented. So to answer your question, yes, if you are going to a large crowded and enclosed environment, such as a long flight or a train ride, or inside a concert hall, which is enclosed environment, please do wear a mask. If you are vaccinated and wearing a mask, you almost can mitigate chances of being infected from another infected individuals who may be asymptomatic. That individual even doesn't know that he or she is infected. And if you are sitting next to that individual, if you are wearing a mask, you can protect yourself being exposed to the viral particle over 75% level if you are wearing a KN95 mask. So answer is yes. In public places, particularly in confined area, if we are staying for an extended period of time, a simple KN95 mask wearing will mitigate, protect us being infected or at least being exposed to the viral particle at the highest level, over 75% level. And if we are vaccinated, our protection even goes up.
Wonderful, thank you. Are there any other questions? Feel free to type them in the Q&A box.
Let me open it up. I am not seeing any other questions.
Well, thank you so much, Dr. Mandal. That was a fantastic and absolutely enlightening presentation. We really appreciate you sharing your expertise and your time again with us. And thank you also to everyone who participated. Again, everyone who registered will receive Dr. Mandal's slides and the YouTube link in the coming days. And we hope you'll join us for future Roadrunner Resource Series presentations. Thank you all and have a wonderful evening.
Thank you.
Good night.
Bye-bye.